Hello everyone, welcome to LKG. So today we are going to discuss about the continued part of the chapter, that is TS of Anther and Microsporogenesis. So let's get started. So in the TS of Anther, we can see 4 wall layers. Let's discuss about the 4 wall layers in detail. So the outermost wall layer is known as epidermis. Epidermis is the first wall layer. It is outermost and it is single layered. It is made up of only one layered cell and it is protective in function. Okay, so epidermis is outermost layer, single layer and protective in function. So the second layer is endothecium. So endothecium is also a single layer and it has fibrous thickenings. And this fibrous thickening is due to the deposition of a chemical which is known as callose. So callose is hygroscopic in nature, means callose have the ability to absorb the moisture. Because of this, the cells of the endothecium will swell, and because of the swelling, the cells will rupture. So this rupturing of anther is nothing but dehiscence of anther. So the endothecium also helps in the dehiscence of anther. Dehiscence of anther. Okay, next the third layer is known as the middle layer. So it is the third layer and it is 1 to 3 layered thick. It is made up of 1 to 3 layers and the cells of the parenchyma, the cells of the middle layer are parenchymatous. It is made up of parenchyma cells. Okay, and on anther maturity, the middle layer will degenerate. So it will degenerate on anther maturity. Hence, it also indirectly involved in the dehiscence of the anther. So the first 3 layers, epidermis, endothecium and middle layers, are involved in the protection and dehiscence of the anther. The fourth layer, it is a special layer in structure as well as the function. Fourth layer is tapetum. This is the fourth layer. So why it is special in structure is because tapetum has dense cytoplasm. Not only dense cytoplasm, it has 2 or more nucleus. The cells of the tapetum has 2 or more nucleus. Hence, it has diploid or polyploid cells. The function of tapetum is, it is a nutritive layer. It provides nourishment. Nourishment to whom? It provides nourishment to developing pollen grains. So its function, it is a nutritive layer and provides nourishment to the developing pollen. Apart from this, this also has small granule which is known as Ubish body. What is this Ubish body? So this Ubish body is made up of an important chemical, highly resistant chemical which is known as sporopollenin. So later the sporopollenin is used up in formation of the outer layer of the pollen grain, that is exine. Okay, so these are the 4 wall layers of the anther, that is epidermis, endothecium, middle layer and tapetum. So next, inside the tapetum, you all know there are microsporangia, and inside the microsporangia there is a special type of tissue which is known as sporogenous tissue. So here you can see inside there is sporogenous tissue. So each cell of the sporogenous tissue is a potential pollen. Means each cell of the sporogenous tissue can give rise to a pollen grain. Hence there is a special name for each cell of this sporogenous tissue. It is known as pollen mother cell. Means it will give rise to pollen grain. Hence the name is pollen mother cell, or it is also known as microspore mother cell. So remember, pollen grain and microspore is same. Okay, pollen mother cell or microspore mother cell. Now let's see the microsporogenesis. Okay. What is this microsporogenesis? So as I told you, see, this is the pollen sac. Inside the pollen sac there are lots of sporogenous tissue. And what is each cell of sporogenous tissue known as? Each cell of the sporogenous tissue is known as pollen mother cell. So let me draw a pollen mother cell. This is a pollen mother cell, or PMC we call it as, or microspore mother cell, MMC. Okay. So this pollen mother cell or microspore mother cell will undergo meiosis. It will undergo meiosis, which is a reductional division, and it gives rise to 4 cells which are not completely separated. Hence this 4 cell is nothing but the pollen tetrad or microspore tetrad. So further, when the anther matures, this pollen tetrad will separate and each cell becomes a pollen grain. So now how will you define microsporogenesis? The process of formation of pollen grain or microspore from microspore mother cell. So this is known as microsporogenesis, where the pollen mother cell will undergo meiosis and form pollen tetrad or microspore tetrad, and when the anther attains maturity, the pollen tetrad will separate and each will become a pollen grain. Okay, so that's all for today. Hope you understood the topic. Please subscribe to the channel and share with your friends. Thanks for watching.